Today, FDA approvals in pancreatic cancer, a diagnostic for blood cancer detection, and alopecia, impressive findings in a phase 3 melanoma study, and a European approval in non-small cell lung cancer. Welcome to Enclave News Network. I'm Gina Columbus. In pancreatic cancer, the FDA has approved panitumumab in combination with FOLFOX as a frontline treatment for patients with RAS wild-type metastatic colorectal cancer. In addition to the frontline approval, the agency also cleared panitumumab as a monotherapy for RAS wild-type mCRC following prior treatment with fluoropyrimidine, oxaliplatin, and irinotecan-containing chemotherapy. For the determination of RAS status, specifically exons 2, 3, and 4 of KRAS and NRAS, the FDA also approved a multi-gene next-generation sequencing-based companion diagnostic. The expanded RAS indication was based on an improvement in overall survival in the prospective phase 3 201-00007 study and in a retrospective biomarker analysis of the phase 3 PRIME study. The addition of panitumumab to FOLFOX improved OS by 5.6 months for untreated patients with mCRC in the PRIME study. Additionally, panitumumab improved OS by 3.1 months versus best supportive care in the 201-00007 study for patients with chemorefractory RAS wild-type mCRC. The FDA has also approved Clear-L Lab multicolor reagents T1, T2, B1, B2, and M for the detection of chronic leukemia, acute leukemia, non-Hodgkin lymphoma, multiple myeloma, myelodysplastic syndrome, and myeloproliferative neoplasms. This approval represents the first from the agency for immunophenotyping on a flow cytometer. The approval was based on the efficacy of the screening panel versus alternative methods for the detection of leukemias and lymphomas in 279 samples at 4 clinical sites. The findings from the Clear-L Lab reagents aligned with standard methods in 93.4% of the samples. Additionally, the diagnostic correctly detected cancer in 84.2% of samples. These reagents can be used on peripheral whole blood, bone marrow, and lymph node specimens. Each of the Clear-L Lab reagents is specific to CD45, which is expressed on a variety of leukocyte populations. In addition to CD45, the tests provide qualitative results for several other factors associated with hematologic malignancies. The T1 reagents illuminate CD2, CD56, CD7, and CD5. T2 is specific to CD8, CD4, and CD3. The B1 test detects kappa, lambda, CD19, and CD5, while B2 is specific to CD20, CD10, CD19, and CD38. The M test detects CD7, CD13, CD34, and CD33. The FDA has also expanded the indication for the DigniCap cooling system to be used for alopecia reduction across all solid tumors. The device had previously been approved in 2015 solely for reducing severe hair loss in patients with breast cancer. The initial breast cancer approval was based on an analysis of 122 women with stage 1-2 breast cancer who were undergoing alopecia-inducing chemotherapy. Results of the open-label, non-randomized study showed that more than 66% of the women who used the device lost less than 50% of their hair. None of the patients in the control arm met this milestone. The cooling cap circulates cold liquid through a silicone cap that is worn on the head during treatment with chemotherapy. The cap is connected to a unit that distributes the cooled liquid that is controlled using touchscreen prompts. A second cap made of neoprene holds the initial cap in place and prevents loss of cooling. The near-freezing temperatures are intended to constrict the blood vessels in the scalp, making it difficult for toxic chemotherapy agents to reach and harm the hair follicles. The cold also decreases the activity of the hair roots, which slows down cell division and makes them less affected by chemotherapy. In melanoma, adjuvant treatment with nivolumab was found to extend recurrence-free survival versus ipilimumab in patients with advanced disease. Data from the phase 3 CheckMate 238 trial have not been released, but the manufacturer of both immunotherapy agents, Bristol Myers Squibb, reported that 3 milligrams per kilogram of nivolumab met its primary endpoint by demonstrating superior RFS compared with 10 milligrams per kilogram of ipilimumab in patients with stage 3B, C, or stage 4 melanoma who are at high risk for recurrence following complete surgical resection. In the ongoing CheckMate 238 study, 906 patients were randomized to receive either 3 milligrams per kilogram of nivolumab every 2 weeks or 10 milligrams per kilogram of ipilimumab every 3 weeks for 4 doses, then every 12 weeks for 1 year or until documented disease progression or unacceptable toxicity. Complete findings will be presented at a future medical meeting. The European Union has approved the use of suritinib as a first-line treatment for patients with ALK-positive advanced non-small cell lung cancer, making the ALK inhibitor now available for patients in all 28 EU member states, plus Iceland, Liechtenstein, and Norway. The approval is based on findings from the phase 3 ASCEND-4 trial, in which suritinib was associated with a 45% reduced risk for death compared with chemotherapy. Additionally, the median progression-free survival benefit favoring suritinib was 8.5 months. The EU's Committee for Medicinal Products for Human Use issued a positive opinion regarding first-line suritinib in May. Also in May, the FDA approved the drug for use in the same patient population. This week, Dr. Daniel Petrolak of Yale Cancer Center discussed his appreciation for being named a 2017 Giant of Cancer Care in genitourinary cancer.
My greatest accomplishment was helping to get Taxotere approved for castration-resistant prostate cancer. And there's a actually interesting little story that goes behind that. So in 1996, we were looking at taxanes in the laboratory with prostate cancer cells, and we saw that Taxotere combined with estramustine had some activity that we really wanted to pursue. And so we designed a phase 1 trial to combine the 2 different drugs, and we sent it to the company that was making the drug at the time, Rompalonc, and they said, well, we've got good news and we've got bad news. The good news is that we can give you drug, and the bad news is, unfortunately, we can't fund it. So we came up with our own funding, and then I remember very, very well treating the first patient who didn't respond, and I said to myself, gee, you know, we put all this effort in and look what happened here. But then the next 5 or 6 responded. And we presented our data at one of the SWOG meetings, and Dave Crawford was really a great advocate. We moved forward with the phase 3 trial, and then the SWOG 9916 trial accrued patients and then led to, or was one of the supplementary studies that got Taxotere approved for castration-resistant prostate cancer.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.